Hello students, today we will discuss about the enzymes. First, what is the enzyme? Enzymes are biological catalyst. In chemistry, we use the catalyst. Catalyst means which increase the rate of reaction or speed up the reaction, that is the catalyst. So in biochemistry these are called biological catalyst, and these catalysts are proteins in nature. These catalyst or enzyme increase the rate of a reaction by lowering the energy of activation, or it is the minimum amount of energy required for a reaction to occur. Enzyme lowers this activation energy and speed up the rate of reactions. Enzyme catalyze near all chemical reactions or all biochemical reactions taking place in the cells of body. These enzymes are not altered or consumed during the reaction. These enzymes are reusable also. We can reuse the enzymes. As in this graph of energy versus reaction coordinate, we can see that there are two types of reaction. One is the with enzyme and second is the without enzyme. In the first reaction with enzyme, we can see that this is the energy level. This energy is required or activation energy, but in case of without enzyme, the reaction require so much high energy, so much high energy. But with enzyme or without enzyme, with enzyme such energy. So in this graph we can see that in absence of enzyme, more energy as compared to reaction with enzyme is required, or we can say that enzyme lowers this energy for a reactions. Enzyme lowers this energy, so this less energy is for a reaction. So this graph show activation energy without enzyme and energy with enzyme. So by this graph we can see when there is use of enzyme, less activation energy is required. So the concept behind the speed up of reaction is that enzyme lowers the activation energy. Then systematic name of enzyme. According to IUB or International Union of Biochemistry, enzyme has two parts. first part, that is the substrate, name of substrate for enzyme. Second part is type of reaction catalyzed by enzyme, and this second part ends with suffix age, mean second part types of reactions and end with the suffix age. As you can see in example, lactate dehydrogenase, name of enzyme. In this, lactate is the substrate, dehydrogenase, that is the type of reaction. EC number, EC, that is the enzyme commission number. These are 4 digits like A, B, C and D. First, A is the main class, B is the subclass, C is the further subclass, subclass, D is the further subclass. B and C digit describe the reaction, while D digit that distinguish between different enzymes of the same reaction based on the actual substrate in the reaction. For example, alcohol NAD oxidoreductase, EC number is 1.1.1.1, means EC number, that is the 4-digit number. In this, first digit, that is the main class, after second, that is the subclass of first, third is further subclass of second, fourth, that is the further subclass of third, and second and third digit that describe the reaction, and fourth digit that distinguish between the different enzymes of same reactions based on actual substrate. This is the EC number. And EC number or enzyme commissioner number also classified the enzymes into 6 different groups or 6 different class according to reaction being catalyzed or types of reaction. Nomenclature was determined by Enzyme Commission in 1961, with the latest update having occurred in 1992. Hence, all enzymes are assigned an EC number. The classification does not take into account amino acid sequence, that is the homology, protein structure or chemical reaction. This enzyme commission or EC number that classified the enzyme these 6 main class. First class, that is the oxidoreductase. Second class, that is the transferase. Third class, that is the hydrolase. Fourth class, that is the lyase. Fifth class, that is the isomerase. sixth class, that is the ligase. Then first class, that is the oxidoreductase. Oxidoreductase mean all reactions having oxidation and reduction reactions are considered into the class oxidoreductase. Oxidation means addition of oxygen, removal of hydrogen, loss of electron. Reduction means addition of hydrogen, removal of oxygen and gain of electron. Transferase, second class. Transferase mean transfer of functional group. Third class, hydrolase. Hydrolase means hydrolysis reaction or breakdown in presence of water. Fourth class, lyase. Lyase mean breakdown in absence of water or elimination reaction to form double bonds. Isomerase. Isomerase mean all isomerization reactions or bonds rearrangement reactions. Ligase. Ligase mean bond formation reaction or this couple with ATP synthesis. ATP hydrolysis, breakdown of ATP. Oxidoreductase, it performs redox reaction by utilizing electron acceptor such as NAD in dehydrogenase, H2O2 in peroxidase and oxygen in oxidase reactions. So oxidoreductase means all reactions having oxidation and reduction, means addition of oxygen or removal of hydrogen, that is oxidation, or addition of hydrogen, removal of oxygen, that is reduction, takes place into the class oxidoreductase. Second class, that is the transferase. Transferase mean transferring of groups like amino group, phosphoryl group, carboxyl group, carbonyl group, etc. between the different substrates. Example of transferase: kinases, transaminases, phosphorylases. Then third class, that is the hydrolase. Hydrolase mean hydrolysis or reactions in presence of water. It performs reaction by adding water which breaks the bond, like phosphodiester bond is broken down by enzyme phosphodiesterase. Amide bond is broken down by enzyme peptidase. Fourth class, that is the lyase. Lyase means breakdown in absence of water. It have groups like CO2, NH2 are either added or removed from the substrate. Example: aldolases, synthases and decarboxylase reactions. Fifth class, that is the isomerase. Isomerase mean rearrangement. Isomer mean same molecular formula but different structural formula. It catalyzes the rearranging the compounds forming isomers. Example: epimerases, mutases and racemic mixture. Sixth class, that is the ligase. Ligase means form new bond by utilizing ATP. Example: carboxylases and synthases. So these are 6 main classes of enzyme: oxidoreductase, transferase, hydrolase, lyase, isomerase and then ligase. Then mechanism of enzyme action. In mechanism of enzyme action, that is, what is relation between the enzymes and substrate. Enzyme is a big molecules and enzyme has a particular site where substrate bind. This particular site is called active site. Where substrate bind and substrate, it is a small molecule as compared to enzyme. So enzyme is a big molecules and each enzyme has a particular site that is called active site where substrate bind. First, substrate bind to the active site of enzyme and there is formation of enzyme substrate complex, and then this enzyme release the product and remain itself as such. You can see here this substrate, it is converted into the product and enzyme remain as such. There is no effect on the enzyme. Substrate bind to the enzyme and is converted into the product. This is the mechanism of action of enzyme, and to explain the mechanism of action of enzyme there are two hypotheses. First is the lock and key hypothesis or lock and key model. Second is the induced fit hypothesis. First is the lock and key hypothesis. Mean imagination. Suppose we have to remember anything, we make the picture of that object in our mind. So first scientist Emil Fischer, he gave lock and key model or lock and key hypothesis. He compared enzyme and substrate with lock and key. He compared lock as an enzyme and substrate is a key. Loc is enzyme, key is the substrate. Loc is a big molecules, like enzyme is a big molecules. Loc has a particular site, like enzyme has a particular site, where substrate or key bind and it is converted into the product. So first, loc and key model. According to loc and key model, enzyme is a loc which has active site where substrate bind and there is formation of first enzyme substrate complex, and then substrate is converted into the product. And according to loc and key model, active site has a rigid shape, means solid in shape or not changeable. Only substrate with matching shape can fit, means like each loc has a particular key, like each enzyme has a particular substrate, or each enzyme is for particular substrate. Substrate is a key that fits the loc of active site. The drawback of this model is that means each loc has a particular key, like one enzyme is specific for one substrate, but this is not true in case of enzyme and substrate, because substrate site or active site on enzyme, that is some flexible that can change the shape. So second hypothesis or induced fit hypothesis was given by Daniel Koshland in 1958. According to induced fit hypothesis, binding of substrate induced a conformational change in the active site of enzyme, and in addition enzyme may distort the substrate, forcing it into a conformational similar to that of the transition state. You can see here, that is, there is a conformational change in the active site of enzyme. Then, as we have discussed, enzymes are protein in nature. Some enzymes require non-protein part which help the enzyme for a reaction to catalyze, and these compounds which help the enzymes are called activators. These activators are generally called prosthetic group, and these are of two types: cofactors and coenzymes. Cofactors, these are non-protein part. These are small or inorganic compounds and ion. Example like magnesium ion, potassium, calcium, zinc, iron, copper. These help in enzyme. Example: iron in hemoglobin, magnesium in chlorophyll. Second, that is the coenzymes. Co means attach. non-protein part, organic molecules. These bind temporarily or permanently to enzyme near the active site. These coenzymes are generally vitamins or its derivatives. Example like NAD or niacin, vitamin B3, FAD, riboflavin or B2, vitamin B2, coenzyme vitamin B5 or pantothenic acid. First, this cofactor, that is non-protein group, needed to activate apoenzyme, that is the protein part. Apoenzyme plus cofactor, that is called holoenzyme. Cofactor like cobalt, non-protein part, which help the enzyme. Second, coenzymes. These are organic molecules that temporarily bind to apoenzyme in order for fit into the work. These apoenzymes are vitamins, and these serve as intermediate carriers of small molecules from one enzyme to another. These are vitamins or its derivatives. A single coenzyme molecules may associate with different enzyme at different times. Then we will also discuss one term, that is the isoenzyme. Isoenzyme mean multiple forms of an enzyme which differ in physical and chemical properties and catalyze the same reaction as an enzyme. Isoenzyme means multiple form of an enzyme which catalyze the same reaction but their location is different. Isoenzymes are produced by single gene and may result from more than one gene. Isoenzyme can be separated by heat inactivation, chemical inhibition or electrophoretic techniques. Example like hexokinase and glucokinase, they are isoenzyme. They catalyze the same reaction, that is conversion of glucose to glucose 6-phosphate phosphorylation. But hexokinase, that is present into the muscle, glucokinase, that is present into the liver. Okay, so today we have discussed enzyme classification and cofactors, coenzyme and isoenzyme. Thank you.